So, welcome again to the North Carolina Aquatic Data Hub's database step-by-step webinar. Um, Stop sharing my screen here. So, today we're excited to share with you a how to use this database. We'll give you a little quick background introduction beforehand. But it's mostly about how to use this. You will find other webinars that we've recorded that are more introductory in nature on our website, which I will share right now in the chat for those of you here live. Anyway, my name is Christy Perrin. I am zooming in from Raleigh here in Raleigh, North Carolina with the Water Resource Research Institute and North Carolina Sea Grant. My co-presenters today, actually the main presenters, are Caitlin Elliott with the Haw River Assembly. You can see her, George Santucci with the town of Boone, and Anne Marie Traylor with the Environmental Quality Institute, or EQI, and Cam McNutt with North Carolina Division of Environmental Quality, Department of Environmental Quality, Division of Water Resources. So those are my co-presenters. I'm going to kick us off with a quick story about why we're here. And this is local to where I am. WRI and Sea Grant are both located at NC State's campus here in Raleigh. We are in the Walnut Creek watershed. So that is our eco address here. And one of the ways we support our watershed is to host a small stewardship corps of community members who support stewardship and community engagement, watershed improvement in the Walnut Creek watershed. And it was not long ago when some colleagues of ours, Professor Angela Allen, who is now a part of this Aquatic Data Hub group, and our riverkeeper, Sam Kropp, approached us and said, well, we'd like to apply for a grant to do some community monitoring in one of these creeks in the Walnut Creek watershed. So we asked our stewardship corps members, community members, if they wanted to be involved, if they wanted to help collect data. And a few of them were interested. Angela and Denise and Alina were all curious and interested, but they wanted to know, even though we were going to give them stipends, they wanted to know what was going to happen to the data. How was this data going to be used? And who was going to see it? And they were very adamant that they wanted to know that this data was going to have a use, it's going to have a purpose, and it was going to have a place where people could see it. So that's really what we're about here, is trying to help organizations make their data available. Okay, so now that we've done that, I'm going to give you a quick little overview of the Aquatic Data Hub. Okay, so this is a group, this is a collaborative group that got together, I would say about, give or take, 10 years ago, after a conference session in Asheville about community monitoring. There was interest from participants in finding ways to better support community monitoring across North Carolina. So this group got together and hosted a summit in Chapel Hill at the Botanical Garden there, and came up with ideas for how could they support community monitoring. And this led to a grant proposal that New River Conservancy hosted. It led to a collaborative group getting together and coming up with a methods manual, which you'll hear a little more about, other tools and supports for monitoring. We hosted some in-person workshops back before the pandemic, and then created a database, which you will see today. We meet on a monthly basis, and it's a pretty casual group. We take that time to come up with products and projects to support community monitoring. And we also, it's a bit of a peer network. for sharing what we're doing, and also for people who participate to ask questions of their peers. So this is something we do by Zoom. Currently, we've been alternating between Thursdays and Fridays once a month at 10. So if you're ever interested in that, please let any of us know. We have a new form on our on that will be on our website probably this week. It's a new form about how you might want to get involved. Okay, so that's our intro to the Data Hub. And with that, I'm going to turn it over to George Santucci, and he's going to give you some more background about our products. And then Caitlin will do a bit of a how-to with a demonstration of the database. And then Anne Marie will then share some of her own case uses. And then finally, rounding it out, George and Cam McNutt will talk about how to export data to DEQ into the EPA. And we expect that'll take until about 11, and then we'll open it up for discussion. If you have any questions that come up, please put them in the chat. And if it's something that's urgent that we need to clarify in order for you to understand what's going on, we'll get that answered. Other things we might wait until after the demonstration just to make sure that we get through that demonstration. So here we go. I'm gonna kick it off, hand it off to George now.
Awesome. Thank you, Chrissy. Appreciate that. And thank y'all for coming today. Again, I'm George Santucci. I am currently the Sustainability and Special Projects Manager for the town of Boone. My history with the Data Hub goes right back to the beginning. I was the Executive Director of the New River Conservancy. So I am up here in the headwaters of the New, and you saw John post that he's in Blacksburg. The New, of course, starts here in North Carolina, flows through Virginia and West Virginia to the New River Gorge. And if you've never been, please go. It's a beautiful place. But again, thank you for coming. And as you can see, the whole Data Hub, all of our information for the Data Hub is hosted by WRRI on this website here that Christy dropped the link in the chat to. So you can go here to see all kinds of different resources. Christy mentioned some videos that we had already produced. You can see as Caitlin scrolls up that this is where those videos are that you have access to learn more about the Data Hub and monitoring in general. And I am going to move now to the methods manual. So I just wanted to show you where that is on the Data Hub website. And I'm not going to spend a ton of time on this, but because we really want to delve into the database and how to use the database, but it depends on where you are in your journey as an organization and organizing volunteer water quality monitoring or doing it yourself professionally. But we developed this document, the methods manual, to help create a structure for programs and all the different things that you would need to consider as you develop your program. And so you can see these are all the organizations that were part of this since its inception who contributed to this document right here. Yeah, so there's this. If you move on, I'm going to jump through the document just to make sure you get through it quickly. And I wanted to point this part out right here. We use this nomenclature or these icons throughout the document. I just want to make sure y'all had a chance to understand them. We have a habitat, chemical, and benthic. And so as you go through the protocols later on in the book, you'll see these icons at the top, so you'll know which one is what that protocol is meant to be. And then the tiered system, I'll get into that in a minute, but there are also these three different boxes or shapes for tiers. Tier 1, and I think the next slide, Caitlin will go. Oh, no, okay. I'll get into tiers in a minute, but this book does cover, or the manual does cover, all aspects of creating your own water quality monitoring program. So safety being the highest priority, right? So when you choose a site or select a site, it needs to be someplace that someone can go and it be safe, either on public land, road right away, or a bridge, or other things. And then, knowing that there are other, if you scroll down a little bit farther, Caitlin, there are other things to consider. So are there poisonous plants, or are there, is there a wasp nest? So there are other reasons, the stream flow is pretty critical. And so you want to make sure that you're in a spot where that is safe to get into the water and not threaten the volunteers' safety. I think you can keep on. And then when you're selecting a site, beyond the safety aspect of it, it needs to be documented really well. You need to know what that latitude and longitude is for that site. You need to have some nomenclature when you create the site, and we'll get into that a little bit later. But as important as anything here is if you choose to select a site that overlaps with DEQ, DWR's monitoring sites, you should talk about that with CAM. Actually, regardless, if you plan on submitting your data to the state, you should check in with CAM regarding your site locations, because there may be all kinds of reasons why that is either a good or bad site. I mean, obviously, you don't want to be in mixing zones of dischargers. There's just all kinds of and CAM might otherwise suggest a site that's not far away that he would really love to have data at. So it's good to coordinate with CAM. Do you have anything to add there, CAM?
No, that covered it.
Okay.
Contact me is a good idea if you're going to start a program up where you intend to submit data for water quality assessment.
Okay. Cool. Thank you. All right. So there's the tiers. So I'm going to get into this really quick. So we wanted to establish different different levels of water quality data. And so tier 1 is the simplest. It doesn't require a quality assurance performance plan. You can use almost any piece of equipment to report data at the tier 1 level. It is just data, though, that will not be used for any kind of assessment or listing, delisting of a stream. It is just a way to it's the simplest way to get involved in water quality data. And so the state's stream app is that tier 1 data. So you can use their application. It's a phone-based app. And you can just use that to just get started. For people that are just curious, interested, if your organization just wants to start dipping its toe in the water, so to speak, then tier 1 is the best place to start. And it's the least expensive, obviously, and the easiest to do. When you step up to tier 2, then your organization will have to have developed a quality assurance performance plan that is submitted to CAM to be assessed, and he will then get back with you and accept that quality assurance plan or make recommendations to changes to that plan. But at the end of the day, once that plan is developed and approved by the state, it is something you must follow if you want your data to be considered tier 2 data. So that's just another step up. And then you'll see in a minute the methods and the equipment used to collect tier 2 data has a higher tolerance or tighter tolerances on how it measures the data. So it's just a step up. It is a little bit more expensive than tier 1, obviously, and a little more rigorous. So then tier 3 is equivalent to what the DEQ does. So you're using all the methods that they use to collect all the data that they collect. And so then therefore that data is used for assessment purposes and for listing, delisting of streams on 303D lists. So that is the highest level, and it typically requires either a partnership with a lab where you're submitting your data to a lab for assessment, or you're using digital equipment, YSI meters and other things out in the field, and you're doing all the necessary maintenance on that equipment to make sure that it's collecting the data with the highest quality. Cool. Okay, we can go on. So then this next table here shows each has the tier 3 protocols, and then each methodology or parameter to be collected and what's different between those parameters as you move between the different protocols. So. And I just wanted to show you a simple example to show all of what I just talked about is all mixed in. So this is one of the first methods that we have in there. Protocol is a habitat assessment. So you see the oak leaf icon that indicates that it's a habitat assessment protocol. And then this particular protocol will work under both tier 1 and tier 2. Again, the only difference between those two would be whether your organization has a QAP or not. And I think that's all I had, right, Caitlin, for the...
Yeah, I think now the TechSoup stuff.
Okay. So I just wanted to give a high-level overview of the methods manual, so you know it's there. Please go there and download it, check it out for yourself, and let us know what you see and what you think. So before I get into the TechSoup, I just wanted to be clear that the Aquatic Data Hub, actually, can you just throw that up there really fast? Caitlin, sorry. Yeah, so this is the homepage for the North Carolina Aquatic Data Hub. It is a database that is housed in Microsoft Access. Many of you may ask, why is it housed in Microsoft Access? And there are lots of reasons for why that is. I know most folks would love it if there was a cloud-based solution. I am sorry that we were working with budgets that would never allow us to develop anything in the cloud. We're talking about, we were working with tens of thousands of dollars. Cloud-based solutions would far exceed 10 times, hundreds of thousands of dollars to develop a cloud-based solution. The other reason for using Microsoft Access, well, two more reasons. One is that Microsoft is very generous to nonprofits, and you can buy this software for almost nothing. So that was another reason for using Microsoft Access. It was the cheapest solution for NGOs to use. And then the final rationale behind using Microsoft Access is that it is on your desktop. Your data is housed on your machine, in your office, wherever that is, as opposed to it being hosted by some cloud-based service that may or may not continue. We're all hearing some nightmare stories about data on federal websites like the EPA and others being moved or removed. And so if all you did was rely on WQX and the EPA to house your data, who knows what happens in the political spectrum and whether things are going to be there or not. I personally had an experience long ago, and I'm not here to knock Appalachian State University, but Boone, the New River Conservancy had a partnership with Appalachian. We had all our water quality data. We were working with a biology professor over there, and he was hosting all of our data on Appalachian server. And just through miscommunication, the IT department was doing some upgrades and upfits and moving things around, and our data just went away in the process, and we never got it back. It was gone. And so that's when we realized we needed to have a copy of our data in our house at all times. And so the Data Hub allows for that, for you to personally maintain and house your data. Let's go to TechSoup really quick. I know I'm running out of time. Just want to show you really fast how to get Microsoft Access if you don't have it. You go to TechSoup.org, click on Microsoft, tell it that you're human. And scroll on down to the other applications. Click on that 34 Office Applications button. Scroll down to individual applications. There's Microsoft Access right there. It's $91 on TechSoup. If you are not a member of TechSoup, you can see right there, it says new to TechSoup. Scroll back, yeah, right there. You can click join today, and it'll bring up a form for you to fill out. TechSoup has to verify and vet that you are actually a 501c3 nonprofit, because that's all they serve. If you are, so then you can join them. They will check and make sure that you are a nonprofit, and then they will get back to you and say that you have the ability to buy at this discounted price from them. So anyway, that's how you get Microsoft Access. I believe that's the end of my part.
Okay, I'm going to take over now. I'm Caitlin Elliott. I'm the Water Quality Program Manager for Haw River Assembly. And so I'm going to go over how you would set up the database. So I'm starting out with a blank one, and I'm going to go over how you add an organization, how you add people, projects, stations, labs, and sampling events. So first, to get started, we're going to add an organization. So you'll click on Add Organization. Organization code can just be a short name for your organization. So for Haw River Assembly, we can just shorten that to HRA. And then for the full organization name, we're just going to put Haw River Assembly. I don't have a current WQX organization ID, but it would probably also just be HRA. Then you're going to want to fill in your address. And this is also DEQ can contact you if needed.
So if we don't have an organization ID, do we just leave that blank?
You can leave that one blank. We'll get to a next part where you will need to come up with a WQX ID to export it, but for this, you can just come use the same thing as your organization code.
I mean, would that be something that at DEQ would provide us, or is that something that we just make up on the fly?
Cam, do you have an answer for that?
EPA would give you an ID when you register for water quality exchange. If you go through us, then we will assign our own number, but the data are stored differently. So,
All right, thanks.
So then our director is Emily Sutton. We're going to put in her contact information along with our website. And so once you're done with that and you've double checked everything, you're just going to want to click save. And then you're done with that. It closes out that window. So the next step, you're going to want to enter people. Here you can include different roles. So you can have staff, you can have taxonomists, you can have volunteers. So in my case, I'm staff. And so we're just going to put in my name. And then if they have any kind of state qualifications, you could put that in here. I can put in what my role is. So again, I am the water quality program manager. So we're just going to put that in here. I'm going to put in my email. And my phone number. That way, all of my contact information is stored. And so you can also use this part of the database to keep track of all your volunteers and their contact information. So once you've done that, you can just click save at the top and then go back to the main menu or just close out of the person section. So the next part is adding a project. So for our organization, we have a couple different projects. We have our volunteer monitoring program, which we call Riverwatch. We also have our ambient monitoring program. So that's where we do weekly monitoring. So we have a couple different ones, but today I'm going to put in data for our ambient monitoring program. So again, we have that organization code. And if you've already put in your organization information, it will already be right there in this drop down. So then, like I said, we're going to do ambient monitoring. And this is the one I was talking about where it says WQX project ID. Again, when you get signed up for WQX, you will be given one. But for the case of this, if you want to export your data, you're going to have to put something here to get started. So I'm going to call it HRA AMP for short. And you'll want to say when this project started. So 2025, 1. And it doesn't have an end date. So for here, I'm just putting it that it's NA. And then primary investigator would be me. So again, once you add in all those people, you're going to have drop down options. And so right now I only have me as an option, but you can add all of your staff to this. And like I said, your volunteers, if they're the one who's collecting the data. And then you can add in a detailed project description. So for this project, like I said, it's weekly monitoring at 7 sites across the Haw River watershed, typically downstream of wastewater treatment plants. Monitoring parameters include turbidity, specific conductance, dissolved oxygen, pH, and water temperature. You can also leave other comments here about what the data may have. And then if there's a sampling design type, in this case, it would be targeted. And then you're going to want to include information about your standard operating procedures, or the QAPP, if you have one. So in this case, ours is called Haw River Assembly Water Quality Monitoring Quality Assurance Project Plan. And so the start date for that would be 2025-01-01 when this project started. And then if you ever update it, you can put an updated date for that. And then it's likely certified by CAM. So you're going to put CAM's name here, or whoever else is certifying your QAPP. And so the certification date is probably after you submit it. So just want to keep filling that out with that information. The SOP could be different than your QAPP, but in this case, they're the same. And once you get this all filled out, you can click save, and then that will be in there. Okay, so apparently you can't put NAs as the end date. You always find fun things on the back end. So you just leave it empty if the program hasn't ended. So the next part, we're gonna go ahead and add a station. So that's below adding a project. So your station name or ID, this can be whatever you call that site. So for our organization, we tend to call it the stream name and an identifying factor. So whether that be a road, a park, or something like that. So in this case, we're looking at New Hope Creek at Stagecoach Road, and this is in Durham. You can give like a little bit more of a description here. So for the location, I'm saying that it's Stagecoach Road in Durham in case there's other stagecoaches in the watershed. And then we want to say what the stream name is, if it has one. And so this is New Hope Creek. And the water quality exchange location ID, we're just gonna put as New Hope, it's Stagecoach, like we did up here. And then for the latitude and longitude, you're going to put these in, and then you can check them, which is a really cool thing that the database will allow you to do. So when you go down here, it says you can look up the below, but you can also confirm the location with Google Maps. So when you click that, this will pop up and it'll show you your point. So right now I am in the middle of nowhere. So this is one way that you can check to make sure that the coordinates that you put in were correct. So mine were clearly wrong. So I'd want to go back and check them. So I know that these are switched and I forgot a negative. So when we do it again, it should show up at the correct location. And this is just to make sure that you didn't actually accidentally write the coordinates wrong when you were out sampling. Oops. Apparently I did. Oh,
Negative.
negative. Negative. See, that's why it tells you. There we go. Yep. So there's New Hope Creek and Stagecoach. So this is our sampling location. It's an interesting, fun area. there's a waterfowl impoundment right there. And so our sample location is right upstream of it. So that's correct. So we'll go back, and I'm not going to click the look up the step below right now. It takes a second and we don't really have time for that, but it will fill in this information such as the ecoregion level and the HUC 12. So I know the HUC 12, so I'm just going to put that in here. This information you don't have to fill out, but if you do know it, you can go ahead and fill it out. But once you've done this, you can go ahead and click save. So now we're going to go to labs. So if you work with a lab to analyze any of your samples, they should be state certified if you're submitting the data to the state. So they will have a code that's associated with them. So in this case, I'm doing PACE Labs, and they're based out of Raleigh. They're who we do our 5 and 30 fecal sampling. So to get a site on the 303D list for fecal, you must sample 5 times in 30 days. And so we work with them to do that. So I'm going to include their information. And this can be found on DEQ's website, a list of all of the verified labs. And sometimes you have to scroll over to get the last bit of information. And so this is helpful because if you have questions about the data you get back, you can also look here to find what the contact information should be. So once you've done that, you can click save. You can close that. And now the last thing we're going to do is, now that we've added all those things, we can add a sampling event. So right down here under set default values, there's add a sampling event. And the great thing about all the stuff we had done prior to this is it's going to allow us to just click from our drop-down menu. So we're going to do site information for New Hope Creek and New Hope Creek at Stagecoach. The organization is Haw River Assembly, and our project is our ambient monitoring program. And so once you add more programs and more stations, those will also be available in that drop down. And then for the date, we're just going to do today. We can do that. So then we want to click that and add. So once we click add, this will pop up. And so we'll have options to either add event details. So how long it takes to get there. We can do different sample types, which are shown here. Visual surveys, habitat assessment, and water quality. So right now, we're going to do that. So from the drop-down list, there's going to be a bunch of different options. So you can start typing. So we're going to do specific conductance, and there's all of the different possible units in here too. You can also put in the values. So for this, we're going to put 5. And then you can put in here what tier the data is. So this would be tier 1 or 2, depending on who collected it and how it was collected. You can also include that information here. You can include if there was issues with the data. The sample was warm when received as an example. There's it could be below the limit, the reporting limit. You could also be putting in information about a blank. So you can also do dissolved oxygen. And so this is an example where you can have a couple different options. So if we want to do percent, you can do that and say there was like 82%. Another example could be turbidity. So there's a bunch of different options, as you can see as I scroll down. These are all the options that you can add into the database. There are a couple things missing, like trash, but those are things that you could include in comments about the sampling event. So once you're done adding data to it, you can click save and then close out. And then I just want to show you that if you want to go in and edit the data, you would go to add a sampling event. You would want to click all of that information back in there. And then it'll come up with what we had just done. So if you go here, you could go in and edit those and then resave it. So if we look back at our data sheet and it actually said 83, you could go ahead and save that. So that would be the new number. Anne-Marie, do you want to go ahead and share some other ways that data can be entered?
Yes, absolutely. Can I go ahead and share my screen now? All right, can you all see my, here, let me close this. Can you see my database here?
Yes.
Okay, great. Thank you, Caitlin. So one thing I will point out is that when you go kind of back and forth in some of these tabs, I personally get doubled up images for the blue part, but it's just for looks. So don't worry about that. If you close the whole thing and open it up, it's single again. Anyway, I'm Anne-Marie with the Environmental Quality Institute over here in Western North Carolina. And we do stream monitoring. We do chemical monitoring and invertebrate monitoring as well. And so we work with a lot of different partners and have this, 35-year-old project that's been going on, since 1990. So we've got lots and lots of data that I'm really excited about getting into this database. And so I just wanted to show you some ways I see that as being valuable for us. So Caitlin was able to show you guys how to enter the data. And so having done a little of that, I'll just kind of go through. So our organization here is the Environmental Quality Institute, and I have all that information here. We are the lab. We're certified for most of the parameters that we test. And so here's our lab certification number and our contact information as the lab. We have two programs, two projects. We have the Volunteer Water Information Network, VWIN, which is our chemical and physical stream monitoring monthly program, where the samples come back to us at the lab that we test for different analytes. And then we have the Stream Monitoring Information Exchange, or SMIE. And that's probably not going to be like the WQX project ID, but I'm just putting that in as a spacer for now. That project started in 2005, and that's where we train volunteers to go sample macroinvertebrates in the streams. So those are our projects. The people right now are me as the staff, and we have a lot of trained volunteers. And this is something I'm going to have to think about and assess the needs of whoever's using this data, of how important the sampler is, because, over 35 years, some of that data has not been recorded, and also, dozens of staff and interns and hundreds of volunteers have gone into this. So for now, as a spacer, I just have myself and volunteers, trained volunteers. I just put in one station. We've got hundreds of stations, and I'm hoping the next step, the next evolution in this database is to be able to bulk, to batch import
So I should be able to take the data from spreadsheets that I have now, format that very precisely to fit into this database requirements, and then be able to upload it all at one time. So that's what I'm looking forward to have like a long list of our sites. And then, so for now, I just have this site with this these coordinates. This is the part that I'm really excited about, the defaults values. And so you you go through here. You go to the main menu and set default values, and it takes you to this. And this is where we can put in so much metadata about our analyses and how we're sampling, how we're analyzing our SOPs, things like that. So here you can type in or put in which project you're using, what your sampling type is. Is it a visual survey? Is it a habitat assessment, grab samples, macroinvertebrates, photographs? And we can tell what our sampling protocol or what our method protocol is. So here are the different standard methods that we use for our different lab tests. This is our, for our macroinvertebrates, we have our stream monitoring information exchange protocol built into this. You can enter in, some of these different information, and then here's where I can put in like all of our lab tests that we do every month, and some of the field methods that our partners use for the data that gets submitted to us. Our, depth of sampling, how it's preserved, what lab analysis, what lab equipment we use or field equipment. And this is where you put in your data tier, based on whether CAM approves you for it or not. And then down here at the end, at the bottom, you can put in even more information about what your media is. So we're sampling water most of the time in a water bottle. There are all different types of things you could select. Different information that some of your end users may be interested in. Like, we have reporting limits. If somebody came back and said, I need your MDL, well, here's the method detection limit, and you could put that in. And the thing about this is that all that is going to be attached to your data points, which is really the value of this in my eyes.
It
So
keeps you from having to have like 15 different spreadsheets to keep track
exactly.
of stuff. And
Exactly.
especially like what I was showing before, where you had to drop down each one of these, this will keep you from having to do that. Like for turbidity, NTU is already selected.
Exactly. Thank you. So then I would go in here and we have, well, I'll show you this. Here, sorry, I'm getting off track. This is what happens when I don't follow my notes. So here are a couple sampling events I've entered, but as Caitlin kind of mentioned, you can't really go in and edit them here. So what you do to get around that is you go back and you add a sampling event. And as she mentioned, you can just select. The only ones you have to do are the ones that are asterisks. So you can pick like the date and project, and you'll probably end up needing to do your station, as you have a lot of data to go back and mess with. But here's where we have our information about a sampling event. And so this is me going in and putting in data about benthic macroinvertebrate sampling with our stream monitoring information exchange program. So here's the date, the times, who did it, anything you want to put there. Here's that day we went in and did a visual survey, we took photographs, and we did macroinvertebrate surveys. And so if you go in here for the photographs, it says this form isn't ready yet, sorry. But a way to do that is to just add your link, like put it in a Google Photos folder, whatever, get that link and put it here in this additional information or photos section. And then if you do that, it'll take you to know if you can see like that photo that you attached it to. So that's that information. If you had other things that you did that day, like you did a grab sample, you could also put, check that and put information in. So here's our visual survey. You can go through like that day there were showers, there was an other smell and things like that. You can just go in, and so we'll have volunteers fill this out in the field, bring it back to us, and then we can go through and make all these check boxes. And down here, you have a place to put in where the volunteer or staff member who was out there made comment. And so you could say, this person said, no leaf packs found. Nearby property owner told the team that their neighbor has been illegally logging in their forest. EQI passed this message on to Asheville DEQ office, so we can put that information in there for anybody using this piece of data. And then you can also have data entry comments. So like, I may not be the volunteer who was out there doing it, but like I'm noticing something about the data that I want to enter. So I couldn't read the description of water odor, so I made it up from Anne Marie. I'm just making this up. This is not an actual data point. So then we go to anything else you want to do that day. So we're going to macroinvertebrates. So this is our stream monitoring information exchange protocol that's been entered into here. So I'm going to open that up, and it gives us a place. So for this program, we've got 46 different taxa. So it's all these different invertebrate groups, families and groups of families that we break it into. And we do a KickNet, leaf pack, and visual survey at each sampling occasion. And so we'll go through and enter all those abundances of each taxa into our spreadsheet here, or into the form. You can make comments if somebody had a comment like, may have been this quick crawling predator, may have been a fragile detritivore. We put it in a vial and returned it to EQI for verification, things like that you could put in there. And then you're going to say calculate. And what it does is it gives you our site score and rating. So this good fair mirrors the DEQ's, it's trying to mirror DEQ's rating system at like a, anyway, so good fair is basically like average. I don't know, so it also will use calculate a Virginia Save Our Streams and Isaac Walton League scores. I don't know why it's saying cannot be determined, but this one is the one that we use for our program that was developed for us. You can show details below, like taxa richness, number of organisms collected, to where if you only found like 5 things, which has been, it has happened here since Helene, or after like a toxic pulse of something came through, you can say like, oh, well, this rating may not be super accurate because we only had 5 invertebrates in the sample. And then different, EPT and intolerant species and things like that. These event comments and data entry comments are visible here too. So you could continue to enter in as you go through the different data sheets that you're working on. I think that's about all I wanted to show you. I will say that, we work with a lot of different partners. So one of the things our kind of next steps for our organization are, one, to like really get a lot of the data in here, at least the newest years of data into this and finalized, ready to submit to DEQ and CAM. We also will need to be communicating with our partners, because the way EQI works is we've got partners in all different parts of Western North Carolina that are interested in their own counties or watersheds. And so we'll need to be in contact with each other about who's got the data, who's giving it to the state, if they are at all, so that we're not redoing it. So it may be that someone else, like we work with like James Environmental Association, and they have other projects besides these 2 they work on us with. So they may have their own database to keep track of the things that their university partners or their interns are working on. So that kind of communication will be the next step in this process for us here at EQI. That's, I think, all I have.
Awesome.
Great, thanks.
Thanks.
We're gonna turn it back over to George and Cam. I don't know who's gonna speak first, but they're gonna talk about how to export the data.
Right. Caitlin, can you bring up the... So a couple of things, I don't know if you noticed, this has been a ton of information already, and we're to the final step of exporting the data so you can share it with CAM or with the EPA's WQX if you choose to do that. But if you could just scroll up to the top there for one quick second. Just on the side there, yeah. Just scroll up. There you go. So you notice here, because Caitlin's using a blank database, there's a big empty space, but when Anne-Marie was presenting, she had her logo up there. So if you see the third button down, you can customize this thing. So when you're using it in your office, it looks like it's yours and your product, and it is. So you just have to put your logo file in the same folder as the database is and hit that button, and it will automatically pull your logo in and put it up on the screen there. So anyway, all right, Caitlin, let's go do the export. So I'll just talk through it. Cam, please jump in and correct or whatever. So after you enter, so she's now using New River Conservancy's data because they have a ton of data in the database, and so you can see all these events that are in there, and they've been using it for quite some time. And so you see at the bottom there are 3 choices to export your database or data, I should say. And so you can just send it to a spreadsheet if you wanted to share. Say you were working with someone who was doing some analysis for you, or you just wanted to share it in general, you could just export it to a spreadsheet and do that. If you want to export it to CAM, oh, sorry, this is what the spreadsheet looks like. Yeah, great. Thank you.
And it's the same format as like when you download data from WQX.
And I know there's been some questions about WQX. We really wanted to make sure that this database was super compatible with WQX because it is the national clearinghouse for all water quality data. So we wanted to make sure that this database talked and played nice with the federal site. And then, of course, we also wanted it to talk and play nice with our state. So we had to customize it a little bit to make sure we could do both the WQX export and the export in the format that CAM wants to see your data. And I think the cover sheet is probably the biggest thing for CAM that is unique to North Carolina. So did you already do that, Caitlin?
I did, but it was kind of blank, but I can pull it up real quick.
Yeah, so it populates this document with information that CAM wants to see. And if it doesn't fill it all in, then you can just pull it into Word and fill it in yourself, or a PDF editor, and fill out because you may not have entered some of that data into the database, so it didn't know what to put in those fields. So it leaves them blank and highlights that you need to fill those in. But CAM's going to want this along with the table of your data, or tables of your data. Okay. And Cam, if I'm leaving anything out, please jump in and chime in as you see fit.
Yeah,
Did you?
again, if you're planning to do a regulatory data submittal, then I would want to work with you ahead of time because there are some other formatting issues once you do extract your data. So it'd be good to contact me sooner rather than later if you're going to do data submittal. And I put our data submittal webpage in the chat also.
And Kelly, did we do the WQX as well?
Yeah, that was the one I showed
Okay, cool,
here.
perfect. Okay, so
And I will say when you do the WQX, it will separate them out. So this is just the physical chemistry stuff. They also split it out into biological. So you'll end up with a couple different spreadsheets. But yeah, this one's just the chemical, physical.
So Kayla, if you don't mind, please go back to the database. Yeah, the main page. Thanks. All right, so the only other thing I want to say to y'all is that, as you know, you can download this database. We would strongly encourage that you do. Now, regardless, I know Emery mentioned that we don't have the batch import. We've been working diligently on trying to get a batch import system set up for this database. So if you do have 30 years worth of data like Emery has, you're not going to hand enter all that stuff. You have no desire to hand enter all that stuff. So we're working with a programmer to get that done. We're close. But since you've been taught how to use this thing, it is our strong recommendation that you download it and start messing around with it now, even if your data is not. Just start playing with it, because if you're like me, you think you remember all this stuff from a webinar like this, and then you got to mess around with it, and you're like, whoa, wait a second. And what I want to say is, if you do start to enter real data in there, which we again would encourage you to do, and we update the software and it has the import function, no harm, no foul. You download the new version, and then you click on that button right there, import from an earlier. So if you enter a year's worth of data into the database, and then you get this new version that has the import function, you just download the new version from the WRI website, and then you import from an earlier version, and all your old data will just be sucked right into the newer version. So again, I hope you just download what we currently have and mess around with it at your leisure, and even enter real data. And again, it won't be a waste of time to enter real data, because you can just merge the two when we have the import function. So I think that's all I can. Do you have anything else on import
No, I think you covered
or
it.
export, I should say? Cool. Well,
Yes.
I think that wraps it up, Christy.
Is there anything that we need to tell our IT departments for those of us that don't control our own computers?
So when access is set up, there are some security levels that might, depending on your organization. I know Christy has run into this problem at NC State, where they had some security levels set in Windows, or Microsoft Office, I should say, and that prevented the macros that are driving this software from working. So you do need to make sure that you enable in Microsoft Office the ability to use macros.
Sometimes that's as simple as right clicking on the document and going to more properties and saying enable, because it won't be clicked and you just have to click a box.
Great, thank you. So we had a few questions in the chat, which I think were mostly answered. But actually, I got one that I need to share as well. So we're going to start with those questions, and then if there's any other questions that come to mind for you, you're welcome after we go through these chat questions to either raise your hand or put it in the chat. And so let me get down to these. Okay, first one I got, which was not to the whole group. So, is wetland or lake data appropriate to use in this database? And the question did talk hydrology or water quality. I think I know the answer to that, but can you speak to the appropriate use of the database?
Yeah, so any water quality data can be entered into this
Yep.
from any water source, any water body.
Yeah, we don't have hydrology parameters in this database that I'm aware of.
Right.
Yeah. Thank you. Let's see. I see somebody had asked about a current list or map of stations that are contributing data to the state, I'm guessing, and Cam did put this in the chat. If you're interested in looking for that map, we can drop it in again. Let me put it in so it's fresh in the chat. This is a map that'll show you all of the stations that are contributing data to DEQ. Did I get that right, Cam? That's what that's for the last water assessment?
Yeah, those are all the stations that were used in our last water quality assessment, including data that were submitted from various parties. It also includes all data collected by DEQ. So that's marine fisheries, Division of Water Resources for the most part.
Great, thank you. Okay, and we had another question. If lab results are from non-state certified labs or lab spectrophotometers that use EPA methods, are those data allowed? And Cam had responded in the chat, if the data were collected under an approved QAPP without a certified lab, then it would be used as tier 2 for non-regulatory purposes. The certified lab would be required for being considered for, the integrated water quality assessment for actual regulatory purposes of classifying waters as well. Okay, I think that was it for the chat questions. Does anybody have any, you can start with questions of clarification, if anything seemed confusing, you're not clear about, or any other questions that come to mind for you.
For those of us that are new to this program, like what's the difference between like the tier 1, tier 2? Like, maybe y'all talked about this earlier. I came in a couple minutes late, but
That's okay. Yeah, so go ahead.
We covered it a little bit, but it's a little bit nuanced. The biggest difference between the tier 3 data and the others are that we can only use tier 3 data for regulatory decisions. And in this case, for water quality assessment, that is putting a water body on the 303D list based on those data, or it's taking a water body off of the 303D list based on those data. So we have to have a very rigorous data approach and documentation in order to make those decisions, because those are approved by EPA. And the result of putting something onto the 303D list means that the state is then required to develop a restoration plan at some point. So there is a financial stake here. Also, removing it from the 303D list is the same way. You're now alleviating the need for a restoration plan. So because of the high stakes involved there, that's why we require the QAPP and the certified lab. And from my point of view, if data are challenged by some third party, which has happened in the past, or if there's a lawsuit involved, then we can't defend the data unless there's a certified laboratory involved. So tier 2, tier 3 data, we want those to be the same level of data, but we can't use data from a non-certified lab to make those regulatory decisions. We do use it for effectiveness monitoring, especially in areas that are ongoing with restoration. We can use this data to make determinations on whether or not we're improving or getting worse or whatever the case may be. If you look at the data collected by UNC Institute of Marine Sciences, that's Hans Perl's lab, they are not certified for chlorophyll A, but their data are very robust and we can use it for effectiveness monitoring, but we cannot use it to take things off of the 303D list. I think I lost everybody.
No, thank you, Cam. Does that make sense? I can't see people's faces. No.
Yeah, clear as mud.
Yeah. There's also, Charles, we also have dropped our website in the chat for the data hub, and there is a brief explanation of those tiers right on that page. I'll drop it in again. So it's
also, are there any grants or anything available for getting equipment to get started, or is that pretty much on, or are we pretty much on our own on that?
That is a really great question.
I would say
There's
there are
almost,
no funds from any state agency for that purpose. There may be local foundations and other philanthropic institutions that may support a program like that. I mean, this program, the Data Hub, was originally funded by the Z. Smith Reynolds Foundation. We all put in a collective grant to develop the Aquatic Data Hub through the Z. Smith Reynolds Foundation. Now, they won't fund a small single organization. We had to actually come together as a collective bunch of organizations to inspire them to give us the $70,000 that they gave us to start this thing. But yeah, so I would recommend, depending on the scale of your program, looking at local philanthropy for help with that.
Okay. Thanks. Yeah, we're just a poor little local soil and water district. So.
I have a question along these same lines. So we're a new organization, Deep River Riverkeeper, still in our first year, and we, of course, are raising funds, and we had a donation of 30 cyclopure kits, which allowed us to monitor for PFAS on 2 tributaries of the Deep River, where I did find high levels of PFAS, and I'm conducting round 2. So each tributary has 6 sites, and I am doing 2 rounds of sampling, and then using that to do some sort of source tracking within the 55 parameters that, they offer through that filter. But I know it's not state certified. Is it appropriate to upload tier 1 cyclopure data just for the sake of keeping that data somewhere for people to be able to see? Or, I mean, is that not something that I would put up here as a tier 1?
and Cam probably should answer that. Go ahead.
Yeah, you can submit the data through, I would use it to go ahead and collect the data, and then you could submit it, and I can steer it to the appropriate folks who would be interested in having it.
Okay, great.
Even if I can't use it for water quality assessment, I can still, like our basin planners use the data, and we have in DEQ, we have a whole kind of separate group of folks that work specifically on PFAS, et cetera, that would be interested in having it. Even
Great.
again, they're not making regulatory decisions on it, but it's something they would probably be interested in seeing.
Great, because we're looking to expand with more cyclopure kits because it is relatively affordable. And then we also had samples by Duke Superfund Lab. Are they considered state certified as a lab, or because I know that they provide some data to the feds, and so I don't know if, I mean, we have that data as well, as and what I'm hoping to do is use this data to then write a grant to then go through PACE Analytical, for example, with certified data.
Yeah, Duke does not submit data to me
Oh, they
intentionally.
don't. Okay.
So I have not. They are required on some of their permits to submit certain data, but I know they're certified for many different things, but they have not gone through the process of submitting data to us.
Got you. Okay, good to know.
And just a quick thought about submitting to EPA's WQX as compared to submitting to DEQ. My understanding is if you submit it to WQX, anybody can download that data. It's there for the public to use, which could be a good thing, especially if there's a student that comes along and wants to do some kind of analysis. DEQ, once you submit it to DEQ, it's going for consideration and programming, Cam, right? It's not being posted for people to download, correct?
I usually post it during our public review period so people can look at the data and see what decisions that we made using the data. And I will tell you right now that Water Quality Exchange, the portal, has not been updated since June 3rd. So my guess is there is I know there is not consultant support for it. And so I would hold off on your expectations of loading to WQX for a while.
Yeah, with the federal shutdown, it's probably a good idea. Okay, are there other questions?
Chrissy, I think
I have-
there's a one in the
Oh,
chat there
yep.
about taxonomy. I'm not going to be able to, I might need to lean on Cam and Anne-Marie a little bit on this. But I did want to touch on one thing regarding benthic protocols. So
Well,
the
let's,
SMI,
can we read that question for
oh,
the
yeah,
recording?
sorry.
Because it won't show up.
Is
Thanks.
there any discussion around incorporating Darwin core standards for taxonomy, the taxonomy portion of the database? This could facilitate interoperability with biodiversity databases like GBIF. So when we developed the database, I'm just going to touch on the historic, how we got to where we got there. We knew we had the Isaac Walton League SOS. That was a tier 1 protocol. So that's in there, as you saw. And then Virginia has a modified version of the Isaac Walton League that's a little more rigorous. So we added that protocol in there, and that the ability to calculate the IBI for both of those. And then we were looking for something that was even more rigorous that would fit that tier 2 section. And Anne-Marie's lab, EQI, had this SMI protocol. And once we all reviewed it, we realized that was a more rigorous protocol and met that standard. It does require that you have certain people on staff that can verify the data and train the volunteers, and volunteers have to go through, they'll send them a sample of bugs and the volunteers have to correctly identify all these bugs. So it is a higher level on a more rigorous standard, which is why then it fits into that tier 2. And then of course, tier 3 is beyond almost all of us. I don't know that there's, what are there, 2 people in the entire state, Cam, that can manage a tier 3 program?
Besides DEQ staff, I think there's 3 people that are certified for benthic collections. I know there's a lot of people that probably could be certified that aren't, but
Right.
no, it's a very specialized process.
So then I'm going to admit that I am not familiar with the Darwin core. I don't know if Cam or Anne-Marie are and want to.
I am also not.
I am not either.
So, Paulina, I think that we are going to be talking soon. I think I owe you an email, and so I would love to talk to you more about and pick your brain about this topic when we do that.
Yeah, for sure. And just for the others who aren't familiar with Darwin Core, it is a globally recognized taxonomic standardization, and it's primarily used for digitizing biodiversity data sets. So like museum collections, a lot of federal critter surveys and such utilize this standard, and then that information is interoperable, or using the standard allows your data to then be digested by things like iNaturalist, the Global Biodiversity Information Facility. Yeah, lots of good stuff there. So looking forward to chatting with you next week.
Thanks for bringing that up.
So I have one more like just basic question. So I guess it goes back to like the whole hub idea of it, the data hub. So where I get that this basically the tier 2, tier 3 data. I mean that's for basically regulatory based submissions. So if you want to submit that data to assist with DEQ in their regulatory mission, that's where you're going to be submitting it. But I guess the tier 1, I'm not so sure. It looks like mainly NC Streamwatch. So I guess my question is, how's the tier 1 data being used?
We are currently in the process of developing some phone tablet-based applications for tier 1 data collection. So theoretically, you would not need to use the Aquatic Data Hub if you're just doing a very small program. And it's mostly for educational purposes, but the application does map the information. There's benthos, water chemistry, the habitat. It's all the ones that George is showing you in the manual, but it's just tier 1 data. So we have these apps that we're going to launch that would just automatically submit the data, and it would be used by me to maybe prioritize places where we should be doing tier 3 level sampling, and also to inform our basin planners about potential problems in places where we are not monitoring. And the biggest part of it is so that people working in watershed restoration at the community level can communicate with each other and learn more about what's going on in their watershed. And we will have a map where they can actually go look at that information as well. This is pretty new. Traditionally, we have not had the time or resources to do any processing of tier 1 type of data, but now we're getting there.
That sounds promising.
Yeah, it sounds good.
And we'll launch that through the StreamWatch. Keep an eye on the StreamWatch page. We'll launch those applications through the StreamWatch page and under the North Carolina Community Science.
Gotcha.
Can
Thank
I
you.
also add to that? And this might be a little circuitous, but we've had these kind of grand ambitions over the years of like, wouldn't it be cool if we could do this, or wouldn't it be neat if it could go this direction? And kind of one of the things I thought was interesting early on discussions is also having a way to visualize this data in addition to just entering it and sharing it, through spreadsheets. And so I could see where having your data in this system, and if we were ever able to get to that up next level, that displaying that educational data for schools or community groups or newsletters or whatever could be valuable. And this is another one of the reasons I really like this whole aquatic data hub that you mentioned. Is I just like getting on the calls every month with these guys, and any new people that pop on every now and then, and just hear what everybody's doing, what the new challenges are. And I'm hopeful that we'll still keep climbing the ladder of doing better and better things, but we need more people to get involved and have different skills. Like we all kind of have different skills and different resources and different people that we know to kind of accomplish things. And so I'm going to put in the plug for people to join in our like aquatic data hub meetings, and just, I think that because we're all just kind of volunteer doing this and at this point. And so getting more perspectives and more voices involved and resources, I think, is where this could level up to the next and next things.
Yeah, that's a really good point. I appreciate you bringing that up, Anne Marie. We are all just chipping in our own time as a collaborative, and the more people who are involved, the more perspectives, the more we can do, the better we can do. And this database is one product that we've worked on and continue to work on, but there's a whole slew of other things we have done and could do together, particularly as public funding through state and federal sources continues to get smaller and smaller for monitoring our streams. The community groups are where the slack is picked up and where the opportunity lies. And I think there's probably foundation support out there too, the community foundations, those types of things may be supportive of those types of programs. So
I have one more question.
Yeah.
If I can. So if we were considering, I'm like Charlie, I'm in a soil and water conservation district in
Right.
Brownsburg. And so if we're trying to consider starting a program to help assess streams and water quality, I guess in my point of view, it would probably be more geared towards that tier 2 if it's just, the district staff, and then a separate program would be more tier 1 for just the general public. But if we were to do that, Cam, would we work with you to see if there were certain areas throughout our watersheds that you are actually eyeing that need more assessment and you're not able to. Because that's kind of where I would think of it as a district kind of filling that need. And if there's no need, then it would make it easier to decide on, you know, more of a tier 1 community program.
Yeah, we only monitor about 30% of the named streams in the state, which is probably about 10% of the water bodies in the state, including estuaries in the Atlantic Ocean. So yeah, there's lots of gaps, but given the relationship between Division of Water Resources and Soil and Water Conservation, it'd be a good idea if we were to meet and go through all the expectations, because there is some sensitivity around data being submitted to me that's being collected in agricultural watersheds especially. So we might want to bring Allie Dinwiddie in on that as well.
Yeah, I'd love to meet if it's something that, you know, if it were a good fit for our district. I'd love to try and start some kind of program where we could help fill those gaps. So
yeah,
I'd
I
love to
would
meet you.
really like that.
So, I guess I can try and send Allie a email and
Yeah.
see if we can't coordinate something.
Yeah, especially if there's several of you guys out there that would be interested in doing this.
So, sounds good.
Same here, Elliot. Keep me in the loop on it.
Great. Charles, can I ask what county you're from?
Dare.
Nice. Okay. Great. Yeah, and this is what we're about, is trying to uncover these opportunities to work together. So yeah, our meetings, our monthly meetings kind of move a little bit based on availability right now. I think we've switched between Thursdays and Fridays since not everybody's available on one of those days at 10 a.m. But I dropped a form in the chat. And it will be on our website. It's a new form, so it's not there yet, but it's basically just a Google form for anyone to put in a question or a comment or express interest in being invited to the next monthly network meeting. So, and I see Charlie, you dropped your email. Thank you for that. And so yeah, you can always go in on that and fill out your question or interest in coming to a meeting. With that, any other questions before we wrap up?
Hey, Christy, at the risk of giving us more work, and only because Anne Marie mentioned it a little while ago, data visualization is an unbelievable big deal. And if you really want your organization, I mean, the value of this data is for people to be able to see it in some graphical format, because not everybody can just crunch numbers in a spreadsheet. So I guess I'll ask if there's interest. So there are lots of products. There are an unbelievable amount of products out there, right? NRC used to use a Google product called the Data Studio. I think Google has modified and changed it. I don't even know what it's evolved into, but there are things like Tableau and Microsoft Power BI, and there's just like data viz. And then of course ARC, Esri's mapping software has its own cap. So it's all depending on, some are free, some are expensive, some you can buy again on TechSoup. You can get a discounted version of some of these things on TechSoup. So anyway, long and short, if there's interest, please let us know and we'll start to think through, and we'll talk about it at our monthly meetings too, about how to, if we want to try to do some kind of training or find some experts that can help teach people how to use those data visualization tools. So anyway.
Thank you, George. Yeah, we're open definitely to you know, what we work on next and how we support people. So like data visualization, we've never done a webinar on that, so that could be something in the future. So great. I'm so glad that some of you have found this useful. I hope it's been useful. It may or may not have been what you expected to see, but we were glad you came. Thank you to all my colleagues who did all the heavy lifting of the webinar. George, Caitlin, Anne Marie, Cam, And we hope to see you at one of our meetings in the near future. And drop us a line. Thanks, everyone.
Thanks,
you.
everyone.